So we have been talking about cell adhesion, thinking that cells once attached always remains attached, and all cells always need to be attached. That's not true. Cells sometimes need to make transient adhesion, meaning a temporary adhesion, to perform their function. So let's look at transient cell-cell adhesion. For example, white blood cells, they lead a nomadic life. They are never at one place. They move from one place to another, and they move from one place to another by the bloodstream and from one tissue to another. This necessitates that the white blood cells have a special adhesive properties, and which allow these properties should allow these cells to stick temporarily. They should engage and then disengage from the cells. These properties depend upon, partly depend upon selectins. Selectins are cell surface carbohydrate binding proteins. We have talked about lectins. So selectins are lectins, but they are present on the cell surface. They mediate a variety of transient calcium ion dependent cell-cell adhesion interactions in the bloodstream. I hope you remember that in one of our previous modules, I showed you a video of a white blood cell traveling in the bloodstream and how it gets attached. Now, let's look at that video again, since we have learned so much more, especially about the cell adhesion. So let's look at that video. So here you can see these white blood cells. They are, rather than rushing through the vessel, they are rolling basically on the surface of the blood vessel. So here basically you have two membranes, one membrane belonging to the white blood cell, the other belonging to the endothelial cell, or the cell that is lining the blood vessel. So there are these sticky molecules which are present here. These sticky molecules are basically making the white blood cells stick in a weak fashion, so it just doesn't zoom past through the site where it has to perform a specific task. So first of all, these white blood cells have to be slowed down. Rather than zooming past a site where they're needed, they just very gradually, very slowly roll. So once they have started rolling, these different sticky molecules are holding these white cells in a weak manner. So they're rolling now till they reach the site where they're supposed to interfere, or they have to perform a particular function. So these are the sticky molecules. If you remember, I talked about spectrin once. Here is the spectrin molecule. We have talked about that. So the spectrin molecule is right underneath the plasma membrane. I hope you remember when I was showing you the bird cage. That's when I mentioned it in this particular course. So here, this interaction basically leads to a temporary binding or transient binding. So let's get back to our presentation. This temporary mediation or slowing down of white blood cells is basically dependent upon the selectins. The Lselectins, which are present on white blood cells, P-selectins are present on blood platelets and or also on endothelial cells, cells that have not been locally activated yet by an inflammatory response. So if there's a damage to the tissue, there's an infection, or if there has been a damage, the cells, the endothelial cells will basically start expressing this selectin, and E-selectin will basically bind specific carbohydrate moieties attached to different structures on the surface of the white blood cells, and they will help them slow down. So we have talked about three different types of selectins. L-selectins on white blood cells or the lymphocytes, P-selectins on the platelets, which are part of our blood, and E-selectins on activated endothelial cells. Endothelial cells only make this type of selectin if they sense there has been a damage in the neighborhood. Each selectin is a transmembrane protein with a highly conserved lectin domain, which binds to a specific oligosaccharide carbohydrate moiety of another cell. So this is the selectin domain, which will bind the carbohydrate moiety. This is the transmembrane domain, which is embedded in the plasma membrane. This is totally entirely the extracellular domain, and this is the cytoplasmic domain of this molecule. Lselectins act as a homing receptor. So Lselectin, again, they're present on the lymphocytes or the white blood cells when they enter the secondary lymphoid tissue. So because they have the Lselectin, the white blood cells have Lselectin, ligand for the selectin, or the molecule which it sticks to, is present on endothelial cells, and it will bind, these white blood cells will bind the endothelial cells in the secondary lymphoid organ because they are expressing the Lselectin. So this basically allows the lymphocyte to stay in the lymph nodes or the lymphoid tissue, so they can basically be activated. So these selectins facilitate the entry of WBCs in the secondary lymphoid organ. At the site of inflammation, endothelial cells express selectins which recognize the oligosaccharides on the white blood cells and platelets, flagging the cells down to help deal with the local infection. So I will just basically draw this. Here's, for example, a secondary lymphoid tissue. This basically has the sugar molecules which can be recognized by white blood cells which have a selectin which will bind this particular type of carbohydrate. So the lymphocyte will allow lymphocyte to spend time in the lymph node. That's where a lot of our immune cells get activated, for example. At the site of, this is again the white blood cell. At the endothelial cells, at the site of infection, endothelial cells basically will express selectins. These selectins will bind a carbohydrate moiety present on the surface of the lymphocyte. So it works in the reverse. In one tissue, it's working in one way, and the other tissue, it's working in a different way. Naive T lymphocytes, which have not yet been activated, they have to yet encounter the specific antigen by the APCs, or the antigen presenting cells. They enter the lymph node and they encounter their antigen via APCs, and then they also have to be exit secondary lymphoid organ. Now again, please appreciate that this interaction has to be temporary, because we don't want the white blood cells to stay there permanently. Central memory T lymphocytes, which have encountered the antigen, they express Lselectin to localize in the lymphoid organs. So these memory cells, which we have seen how they're produced, they produce Lselectin so they can live in the lymphoid organ. Effector T cells, they do not express selectin. So remember, effector T lymphocytes, if they expressed Lselectin, they will be bound in the secondary lymphoid organ. They have to move and find the location where there has been an incident, or there has been damage, so they can perform their particular function.